First, we will start with a coherent-based optical microscope, which has a laser that is the illumination source, an objective nose piece in which 5 microscope objective lenses with different magnifications can be attached, a tube lens with fixed focal length, and finally a digital camera to record images. To convert the optical microscope into a polarization-sensitive system, it is necessary to add extra optical elements. In our case, the laser used is elliptically polarized, so at the exit of it, we place a depolarizer to have randomly polarized light. Just after the depolarizer, we insert a calibrated linear polarizer in which we know the direction of each transmission axis. In our case, we have chosen that each transmission axis is located horizontally from right to left. Once the calibrated linear polarizer is set up, we insert a linear polarizer that is the closest to the camera. The linear polarizer is mounted on a motorized mount to align precisely its transmission axis. Connect the motor to the computer to determine the orientation of its transmission axis. Now, rotate the orientation of the linear polarizer until we find the orientation in which the intensity reaching the sensor is minimum. At this orientation angle, the transmission axis of this linear polarizer is at 90 degrees with respect to the reference, which is the calibrated linear polarizer inserted after the polarizer. Once we have determined the transmission axis of the linear polarizer, remove the calibrated linear polarizer. The next step is to insert a second linear polarizer closer to the illumination source. Using the software of the motor, rotate the transmission axis of this second linear polarizer until the intensity reaching the camera is minimum. Now that both linear polarizers are aligned, it is time to insert the quarter wave plates in the microscope. Firstly, we insert the quarter wave plates closer to the illumination source. As before, we rotate the orientation of the quarter wave plates in order to find the minimum intensity of the light arriving to the sensor. However, for the quarter wave plates, there are 4 orientation angles that produce an intensity minimum. These orientation angles are 0, 90, 180, and 270 degrees with respect to the calibrated linear polarizer. Therefore, it is necessary to check that the phase axis of the quarter wave plate has been aligned with the reference axis of the rest of the system. The phase axis of quarter wave plates is usually marked on the mount with a white line. In this case, one can see that an intensity minimum is found when the phase axis of the quarter wave plate is crossed with respect to the reference transmission axis. So we must rotate the quarter wave plate by 90 degrees. Insert the second quarter wave plate within the objective lens and the linear polarizer that is the closest to the camera. Repeat the process to align the phase axis of the second quarter wave plate. Remember that its phase axis must be parallel to the transmission axis of the calibrated linear polarizer. The last step is to rotate the linear polarizer that is closest to the camera by 90 degrees. Now the optical microscope is ready to measure the polarimetric properties of biological samples.